After the food has been digested, absorbed, and stored, glucose levels begin to drop due to the body metabolic needs. For example, skipping meals or sleeping overnight drop glucose levels in blood. At the moment, in the liver, glycogen is broken down to release glucose by a process called glycogenolysis. When glycogen and blood glucose reserves are low, a metabolic shift happens to save glucose for the brain, which is unable to use fatty acids for energy. This shift involves beta oxidation of fatty acids to produce ketone bodies in the liver. Glycogenolysis. Glycogenolysis is the breakdown of glycogen. In response to a drop in blood glucose concentration, the pancreas releases glucagon. Glucagon stimulates the glycogenolysis in the liver and muscle cells. Glycogen branches are catabolized by a sequential action of enzymes. These enzymes remove glucose monomers from glycogen, and as a result, glucose is released by the liver. As a consequence, there is an increase in blood glucose. This glucose will be used by the peripheral tissues as well as the brain. Due to the demands of the tissues and organs in our body, blood glucose levels must be maintained in the normal range of 80 to 100 milligrams per deciliter. Here's glycogen in the liver, and through the action of enzymes, we are going to get glucose. And this process is called glycogenolysis. Lipolysis. Lipolysis is the metabolic pathway through which triglycerides are hydrolyzed into glycerol and free fatty acids. We can see triglycerides right here, and they're going to be broken down into fatty acids and glycerol. Lipolysis is used to mobilize stored energy during fasting or exercise, and usually happens in the liver and the adipose tissue. The most important regulatory hormone in lipolysis is insulin. Fatty acids are slower than glucose to convert into acetyl-CoA, as first it must go through beta oxidation. As we can see in here, fatty acids, before they are turned into acetyl-CoA, they have to go into beta oxidation. Beta oxidation. Beta oxidation is a chemical process used to produce ketone bodies. The liver produces ketone bodies by breaking down fatty acids and ketogenic amino acids. The production of ketone bodies, acetoacetate and beta-hydroxybutyrate, is called ketogenesis. The liver itself cannot utilize these molecules for energy, so the ketone bodies are released into the blood for use by extrahepatic tissues like the brain, skeletal muscles, heart, and kidneys. On this side of the figure, we can see the liver, where we can see the different chemical reactions to form acetoacetate, which is a ketone body. Then, the liver is going to release beta-hydroxybutyrate into the blood. Extrahepatic tissues is what is being displayed on this side of the figure. For example, the brain. So the brain will take the beta-hydroxybutyrate, which is a ketone body, and through a series of chemical reactions is going to get acetyl-CoA. The formation of acetyl-CoA from ketone bodies is going to be called ketolysis. Acetyl-CoA will enter the Krebs cycle, and the Krebs cycle will continue with the electron transport chain for ATP production. The brain, the heart, as well as other organs produce ATP this way in situations when there is carbohydrate and protein insufficiency. For example, when we are fasting or when we are sleeping. Since there are no carbohydrates and no proteins, then in this situation, the only source available for ATP are going to be fatty acids, and then the fatty acids will have to go through beta oxidation in order to be a source for ATP production. Ketone bodies, such as beta-hydroxybutyrate, are always present in blood in little amounts, but they are always present, and that is considered physiological ketosis. In physiological ketosis, normal regulation of ketone bodies production is maintained, and the blood pH is also maintained. However, in ketoacidosis, there is a failure of the normal regulation of ketone body production. Both acetoacetate and beta-hydroxybutyrate are acidic, and if levels of these ketone bodies are too high, the pH of the blood will drop, resulting in ketoacidosis. Ketoacidosis is the pathological state of uncontrolled production of ketone bodies that results in a metabolic acidosis. Ketoacidosis is known to happen in untreated type 1 diabetes and in alcoholics without intake of sufficient carbohydrates. The result of ketoacidosis is that it will create many metabolic derangements and is a life-threatening medical condition. We can see in the figure that we have an excess of lipolysis, Therefore, there's going to be an increase of free fatty acids to the liver, and this is going to create an increase in ketosis. And if this is not controlled, it may develop ketoacidosis.